recently identified a novel senolytic approach to kill multiple types of senescent cells. His presentation today is Dissecting Primary and Secondary Senescence to Enable Novel Senotherapeutic Strategies. You can read his presentation summary and bio on the website, on the events website. And yeah, let's welcome him on our stage, to our stage. Enjoy the presentation.
All right, thank you everyone. And thank you, Maria, for organizing this wonderful presentation. Today I'm going to talk about mainly on secondary senescence, but I will also talk about generally what senescence is and what the difference between primary and secondary senescence. If you want to see full screens of the slide, you can go to the right bottom corner and make it full screen. Okay. Okay. All right. Let's start with what senescence is. Amit Sharma this morning, he did a very good introduction about the senescence cells, so I don't want to go to details. But for those of you who are not here, or for those of you who want to a brief introduction, when a cell can undergo different kinds of stress, that could be genotoxic stress or mitochondrial damage or telomere attrition or different kinds of stress, the cells will undergo a permanent cell cycle arrest, and these cells are called senescent cells. So the senescent cells are known to secrete a group of proteins called SASP. SASP stands for senescence-associated secretory phenotypes. And the SASP factors are also known to propagate the senescence phenotype to the neighboring cells or even to the far distant cells in a manner of paracrine or juxtacrine senescence. I will come to that point in detail. So generally, senescent cells are non-dividing cells, and they are known to be apoptosis resistant. So they keep accumulating in our body. So when we are young, we might have very little senescent cell accumulation in our body, but gradually we keep accumulating these senescent cells when we get older and older. Now, the important question we are asking here is why senescent cells are accumulating when we age. And we believe there are at least three main mechanisms or reasons for the senescent cell accumulation. One is immunosenescence, by which there is a decline in our immune system when we age. So the immune cells will not be effective on removing the senescent cells, which then lead to the contribution of senescence accumulation. The second reason is, in addition to this, the immunosenescence, the senescent cells by themselves are highly heterogeneous, by which some type of senescent cells, they express a special type of inhibitory ligands on their surface, and they prevent the attack from the immune cells like the NK cells and T cells, and they become resistant to immune-mediated clearance. Third and most important for this presentation is that we know senescent cells, they secrete a group of proteins called SASP factors, and these SASP factors can propagate senescence either in the paracrine or juxtacrine manner. We collectively call this type of cells as secondary senescent cells, and my talk mainly will focus on these secondary senescent cells. So we know that to date there are already a good amount of evidence that shows that secondary senescent cells have a very important role or contribution for senescent cell accumulation across age. For example, if you see this interesting study from Mayo Clinic, they showed that when they inject a mice with senescent cells, where these senescent cells can be detected, the injected senescent cells can be detected by a luciferase receptor reporter, they found that senescent cell injection will lead to excess amount of senescence accumulation in the young mice. And most importantly, what they observed is that they also found senescent cells that are positive for multiple senescence marker, but they are negative for the luciferase receptor, which indicates that these cells are the recipient's own cell that becomes senescent through a secondary senescence manner. So this is an important observation. This recent another interesting observation where they did a single heterochromatin blood exchange between young and old mice. They found that when they exchange blood from old mice to the young mice, they observed a senescence transfer from the old mice to the young mice. Even though they didn't claim that this is secondary senescence, what they found is that when they inhibit the SAS factor production from the old mice, then the old mice blood were not able to propagate senescence to the young mice, which indicates that SAS factors are an important factor in the old blood, which might be responsible for this senescence propagation. So now we know that these senescent cells are generally accumulate across age, and the secondary senescent cells also play an important role in senescence accumulation across age. So the end goal, I think, of research on senescent cells is just to remove the senescent cell accumulation, to remove the cells from our body, because all the senescent cells has been associated with aging or age-related diseases. And for this, it has been already shown that either using a genetic approach, for example, by targeting P16 positive cells, even though we know that this is not really a specific senescence marker, or using senolytic drugs like, for example, the D and Q combination. Uh, Other groups already shown that when we remove senescent cells, it leads to a significant improvement in health span and to some extent lifespan extension. For example, if you see these two mice, the mice on the left side is they are the same age. The left side is without senolytic treatment, whereas the right side is with senolytic treatment. You can clearly see that the one which is treated with the senolytics looks healthier and younger. So removing senescent cells from our body has been shown to be a very promising strategy for aging intervention. So if you have this interesting method of removing senescent cells for lifespan and health span extension, what are the limitations and challenges of the current senolytics, or generally the way we remove senescent cells? One important notice in senescent cells is that they are highly heterogeneous, and because of that, some of the cells are resistant to senolytic treatment. Some senolytics are effective in one type of senescence, other senolytics are effective in other type of senescence. So there is no single senolytic drug that is effective to remove all type of senescent cells in our body. And there is also some issue of the safety of the senolytics that has been in trial to date. And most importantly, and the main focus of today's talk is that we know that secondary senescent cells are important contributor for senescent cell accumulation. However, whether the current senolytics are effective on killing these secondary senescent cells is unknown, because we know that the effect of senolytics is dependent on the cell type of origin or the senescence induction method. And we know secondary senescence is a different type of senescence induction. So we need to know if the senolytics that has been discovered based on the characteristics of primary senescent cells is effective on secondary senescent cells. So because of this, we ask this question that what is the difference and similarity between primary and secondary senescent cells? And then we started this journey by starting just simple senescence induction, primary senescence induction using doxorubicin treatment. And most of the studies that I'm going to present here are in primary endothelial cells, but it has also been confirmed in other cell types like fibroblasts and some other senescence induction methods like irradiation. But most of the talks I am going to focus endothelial cells, and primary senescence was induced by doxorubicin treatment. And then after this, we collect the conditioned media from these doxorubicin treated cells. And using this conditioned media, we induce secondary senescence. So, and then we measure multiple senescence markers. And as you can see here, the red bar is for primary senescence, whereas the green is for secondary senescence. Based on multiple marker, doxorubicin treatment leads to up to more than 90% of cells becoming senescence based on multiple marker, whereas the conditioned media from these primary senescent cells leads to only a maximum of 40% senescence based on multiple markers. So in general, the conditioned media or the substance from the primary senescent cells leads to only maximum 40%, meaning the remaining 60% cells are non-senescent cells. So generally, when we say the secondary senescent cell in a dish, it is actually a mixed populations of senescent and non-senescent cells. And because our aim is to compare primary versus secondary senescent cells, we believe that it is unfair to compare 90% senescence with 40% senescence. So for this, we aim to isolate senescent cells based on surface marker and then do the comparison. So there are a couple of proteins that has been already reported as a senescent cell surface marker. One of the interesting protein is DPP4, which is also known as CD26. A couple of groups already shown that this protein is a senescent cell surface marker in multiple cell types. And we confirmed in our lab that non-senescent cells, they don't really have this DPP4 expression on their surface, whereas senescent cells, they have high level of this DPP4 protein on their surface. That's interesting. So now we ask it, can we isolate senescent cells based on the DPP4 surface expression? To briefly go over the protocol that we used, first we induce senescence, both primary and secondary senescence, and then we incubate these senescent cells with DPP4 antibody, which is conjugated with a fluorophore PE. And then we use a microbead, anti-PE microbead, to stain this DPP4 PE conjugate. And then after that, we run the cells into a magnetic column, into a column which is attached to a magnet. So what will happen here is that because the magnetic bead is bind to the column, so DPP4 positive cells will bind to the column, whereas DPP4 negative cells will flow through. So we collect DP4 negative cells, and then after that, we detach this column from the magnet and then wash the cells and then collect these DPP4 positive cells by hoping that these DP4 positive cells will be majorly senescent cells, whereas the DP4 negative cells might be non-senescent cells. Then after we successfully isolate these DPP4 positive senescent cells in both primary and secondary senescence, and then we confirmed if this isolation enables us to enrich the percentage of the secondary senescent cells. And then here we measure multiple senescence markers. Again, red is for primary senescence, whereas green is for secondary senescence. The others are two non-senescent controls. So as you can see here, after isolation, earlier it was only 40%. Now we able to increase the percentage of secondary senescence up to 85%, whereas the secondary senescence is 90 plus percent. And we don't see any statistical significant difference between primary and secondary senescent cells. And now we believe that there is relatively purer populations of both primary and secondary senescent cells after isolation. So this purer population might help us to really characterize what secondary senescent cells looks like. So then we asked the question, what is the difference between primary and secondary senescent cells? The two important questions we are asking here is that, do the secondary senescent cells have the same pro-survival mechanism like the primary senescent cells? And are the current senolytics really effective on ablating this secondary senescence phenotypes? We know that primary senescent cells or general senescent cells are resistant to apoptosis, mainly because they express several of the pro-survival proteins, specifically the BCL2 family proteins, for example, here are known to be upregulated in different types of senescent cells, and because of this, they are resistant to apoptosis. And several groups showed that if we target these BCL2 family proteins, we can successfully kill the senescent cells with minimal effect on non-senescent cells. For example, if you see this paper on the right side, they use navitoclax, which targets the BCL2 family proteins. It kills up to 80% of the senescent cells. When I say senescence here, generally these are primary senescent cells, with no or minimal effect on non-senescent cells. So this is important observation. So now what we are asking is, do the secondary senescent cells have the same pro-survival mechanism like the primary senescent cells? And for this, we measure the expression level of the BCL2 family proteins in both primary and secondary senescent cells, the BCL-XL, BCL-W, and BCL2. If I take your attention to the section highlighted in red, the first column in the right is secondary senescence, followed by primary senescence. So primary senescent cells, not surprising, they have high levels of these BCL2 family proteins. But for our surprise, the secondary senescent cells, they don't express the BCL2 protein. So then we ask, okay, if they don't express the BCL2 protein, then how about the senolytics that are known to target the BCL2 family proteins? And then we treat both primary and secondary senescent cells with navitoclax and see the effectiveness of this navitoclax on both primary and secondary senescent cells. And as you can see here again, red is primary senescent. There is a significant decrease in the viability of these primary senescent cells after navitoclax treatment, whereas we don't see any significant effect on secondary senescent cells. And the take-home message from here, I think, is that secondary senescent cells have a distinct pro-survival mechanism from that of their primary senescent, and they are resistant to current senolytics. One thing I need to mention here is that even though I showed only navitoclax here, we test several of the current senolytics, and they are not really effective on killing secondary senescent cells. Now, this is an interesting observation, and then we are excited to know more about the difference in similarity between primary and secondary senescent cells. And then to address this, we decided to do RNA-seq on primary and secondary senescent cells. From now on, when I say primary and secondary senescent cells, I have to mention that these are DPP4 positive isolated primary and secondary senescent cells, even though we confirmed most of the phenotypes that we observed even without isolation. So here we isolated both primary secondary senescent cells, DP4 positive cells, and then we run RNA-seq. And then if you see like a very simplified version of our RNA-seq analysis, here on the left, what you can see here is secondary senescent cells, they are clustered together and they are well separated from that of the primary senescent cells. And they have a very different gene expression pattern comparing all the differentially expressed genes in these two conditions. And we also did a 2D PCR analysis, which shows the difference and similarity between different conditions. And what we found here is that secondary senescent cells and primary senescent cells, they are well separated from each other and from their non-senescent controls. And I think the take-home message from here is that in generally, looking the gene expression profile of primary and secondary senescent cells are really different, or they have different gene expression profile from the primary senescent cells. So this is interesting. And we also did several downstream analysis on looking on the details of the pathway and gene ontology terminologies, what makes them different and similar. And one interesting point that we were much interested was, even though we know that generally looking at the gene expression profile, the primary and secondary senescent cells are different, we are asking here, do they have something in common? So we focused our analysis on the genes that are common both in primary and secondary senescent cells. And for our surprise, the top hits in both primary and secondary senescent cells, when we look at the commonly expressed genes, were iron metabolism related genes. If you see here this heat map, the top 3 rows are primary senescent, the bottom 3 are secondary senescent cells. If the top When the top is upregulated, which is blue, mostly it is also blue in secondary senescence and vice versa. So not only the top hits are iron metabolism related genes, but they have the same direction of changes, where similarly either upregulated or downregulated. And we confirmed some of the classical markers of this heat map by using qPCR, and we found that significant number of genes related to iron metabolism are affected in both primary and secondary senescent cells. So this brings another interesting question. If iron metabolism is dysregulated in both primary and secondary senescent cells, do these cells have iron accumulation? And for this, we first measure the total iron level in both primary and secondary senescent cells. On the left hand, the bar graph showed the total iron accumulation in senescent cells. Red is primary, green is secondary. There is high level of iron accumulation, total iron accumulation in senescent cells in general, in both primary and secondary senescent cells. And we know there are two types of iron, ferrous and ferric. And the ferrous iron is also known as labile iron, which is the toxic type of iron. And we were interested to know this toxic iron is accumulated in senescent cells. And for this, we use this specific Ceronox dye, which specifically detects the ferrous iron, not the other type of iron, by applying flow cytometer. And what we found here is that the red one here is primary senescent cells, and green is again secondary senescent cells. The shift towards the right indicates more iron accumulation. So generally, both primary and secondary senescent cells have high labile iron accumulation compared to their non-senescent controls. So it is very interesting to observe that iron accumulation is present in both primary and secondary senescent cells. Other groups also showed that there is iron accumulation in senescent cells, even though they didn't show if it is also present in secondary senescent cells. And most importantly, recently, it's not actually a paper, it's in a bioarchive, it's not published yet. They showed that iron accumulation by itself drives the senescence and senescence associated secretory phenotypes, and it's also important driver for the fibrosis to happen, as it's measured by different fibrosis markers here. So this might tell us, like, at least partly iron accumulation might be responsible for the deleterious effects of senescent cell accumulation in our body. And I think this is very important because iron has been known to be accumulated not only in senescent cells, but also iron is accumulated across age. For example, this study, they measured the iron level accumulation across age in mice at 6 weeks, 16 and 24 weeks. And what they found is there is a gradual accumulation of iron. I'm just showing here the brain, but they also measure other tissues. And there is a generally gradual increase in the iron accumulation in different tissues, at least in mice. So there is iron accumulation in senescent cells. There is also iron accumulation when we age. And we believe now that iron might be an important target specifically for senescence or generally for aging intervention. So what is the role of iron, generally speaking, specifically on the labile iron, because we are mostly interested on the labile iron. We can talk a lot about the labile iron, but today I'm mainly interested on the role of labile iron in ferroptosis. So ferroptosis is a non-apoptotic programmed cell death. And for the ferroptosis to happen, at least three things are required. One, accumulation of this labile iron. Second, lipid peroxidation, specifically the membrane lipid peroxidation. And third, a compromisation in the antioxidant system. So now we know that senescent cells accumulate this labile iron. So if they have high level of labile iron, it will undergo a process called Fenton reaction, which leads to ROS production, and then which further activates lipid peroxidation, specifically the polyunsaturated fatty acids present in the phospholipid of the plasma membrane. And then if this lipid oxidation is not able to prevented by the antioxidant system, then the cells will undergo cell membrane rupture and finally cell death through a process called ferroptosis. Now, okay, we know that several of the genes that are related to iron metabolism are among the top hits from our RNA-seq data. And because we know this labile iron has a role in ferroptosis, we again go back to our RNA-seq data and look if the ferroptosis-related genes were also dysregulated in both primary and secondary senescent cells. And this is the same heat map as previously described. The top three are primary senescent, the bottom three are secondary senescent. So generally, several of the ferroptosis-related genes were dysregulated in both primary and secondary senescent. And then we confirmed this by qPCR. And most importantly, these genes like PTGS2 and CHSC1, which are important driver of ferroptosis. And then the last one, SESL4, which is an important step for the lipid peroxidation, they both are upregulated. So upregulations of these regions tells us that the ferroptosis process should happen, but we know that the senescent cells are resistant to generally death, whether apoptosis or ferroptosis. They are not dying; they are accumulating in our body. So we thought that okay, so if these genes are upregulated, generally these senescent cells are primed for the ferroptosis, but they were not able to execute the ferroptosis process for some reason. And then we hypothesize here that, okay, so can we give these cells some ferroptosis inducer and then activate ferroptosis to kill these senescent cells? And for this, we did a small screening of different ferroptosis inducer drugs. I'm not going to talk about all the screening. Just to show you one of the interesting hit that we found is this ferroptosis inducer drug called FIN56. So we treated both primary, secondary senescent endothelial cells with FIN56 at different dosage. And as you can see here, the red and green line here are senescent cells, primary and secondary senescent cells, whereas the top lines are their respective non-senescent control. So we found out that FIN56 specifically induce cell death in senescent cells, in both primary and secondary senescent cells with very minimal effect on non-senescent cells, especially at the lower dose. And we confirmed this observation in other cell types, fibroblasts, and we also showed in other senescence induction method like irradiation or etoposide treatments. And multiple senescence type, multiple senescence induction methods, it seems that ferroptosis induces specifically ferroptosis in senescent cells, both primary and secondary senescent cells. The last piece of question that we are asking was if the mechanism of cell death by FIN56 treatment is actually through ferroptosis. For that, what we did is first we co-treat these cells with FIN56 in the presence of FER1. FER1 is a ferroptosis inhibitor. So if the mechanism of cell death is ferroptosis dependent, then FIN56 shouldn't kill the cells in the presence of this inhibitor. And that's what exactly we saw here. So alone, FIN56 kills significant number of senescent cells, but in the presence of FER1 ferroptosis inhibitor, it didn't have any significant effect on the viability of the cells. Next, we also measured if the iron, the labile iron, which we know is very important for the ferroptosis to happen, is required for FIN56 mediated clearance of the senescent cells. And what we did was we combined FIN56 with DFO. DFO is an iron chelator, so it inhibits the availability of iron 2, and phenton oxidation will not happen. So through that, it inhibits ferroptosis. And in the presence of this iron chelator, FIN56 was less effective on killing senescent cells. Lastly, we asked if FIN56 induces apoptosis, and here we use a pancaspace inhibitor to inhibit all the apoptosis pathway. And what we found here is that in the presence of this apoptosis inhibitor, FIN56 was able to kill both primary and secondary senescent cells. So combining this data, it tells us that FIN56 induces ferroptosis in both primary and secondary senescent cells, and the mechanism seems to be ferroptosis, but not apoptosis. All right. So just to summarize, what we did was we get endothelial cells, primary endothelial cells from 3 donors, and then we induce primary senescence with doxorubicin treatment, and we use a conditioned media from these doxor-treated cells to induce secondary senescence, and then after that we isolate DPP4 positive primary and secondary senescence cells in order to enrich the percentage of secondary senescence cells to get a purified senescent cells. And then after we did RNA-seq, and we found out that ferroptosis as a potential target for as a potential senolytic target, and we confirmed that at least one of the ferroptosis inducer effectively kill senescent cells with minimal effect in non-senescent cells. The other important thing that we are currently working on is that because we know that these senescent cells have high level of iron accumulation, can we use this as an advantage to remove the senescent cells based on their iron accumulation? For this, we have a preliminary data, but I'm not going to present today because still it is ongoing experiment, but it is a really promising strategy where we collaborate with Adam Ranslow at UCSF, and they enable us to synthesize a drug that specifically targets cells with high labile iron. And we found that approach is really very promising approach where it kills almost all the senescent type with very minimal effect on non-senescent cells. And we are currently also trying if this approach can be applicable in vivo, and we are trying it in 3MR mice model. We have a very preliminary data that showed that it is really promising. I hope I will give you an update in the next presentation. But for now, this is all I have. Finally, I would like to thank you, our own Sense Research Foundation and For a Very Healthy Foundation to sponsor this really exciting project. And finally, thank you very much all the groups. Special thanks to Amit Sharma, wonderful group leader. And the Afe is our scientist. He's around here. You can talk to him if you are really interested about especially immunology and senescence. And Ashley is a master student. She has a wonderful poster presentation. You can talk to her if you are interested about cell surface. So we use DPP4 as a surface marker, but now we have like other very interesting potential surface markers from the phage display analysis and other methods. And talk to Ashley if you want to know more about that. And Mani is a new scientist joined us. He's very interested about aging and Alzheimer's disease. And you can talk to him if you know more about Alzheimer and aging and all those stuffs. Thank you very much.
Wow, Tess, thank you. That was remarkable and fascinating. Thank you so much. I feel really inspired when I listen to our scientists present their work. It renews my enthusiasm and passion for the mission. And I'm very grateful to all of you, for all of you. I don't know if I think you just answered, Tess, the question that was in the chat. You did, right? Because I was backstage and I couldn't hear well.
Let me check the chat.
If iron accumulation can drive senescence in the mouse models, because we have some more questions coming. Can you see the chat, or you want me to read them for you?
Oh, I can see it. So the question is, let me read it. Okay. If iron accumulation can drive senescence in the mouse models described, is there any significant possibility that practices such as iron supplementation in human, especially humans who do not have iron deficiencies, could also accelerate aspects of aging? Interesting question. One thing I would like to mention is that so there are generally two types of iron: labile iron, iron 2 plus, and then the other iron, which is mainly important in the hemoglobin and the transportation system, is iron 3 plus or ferric iron. So I think the problem mainly comes from the labile iron, which is accumulated in senescent cells or generally across age. I don't think it is generally from iron supplementation. So we need to know what type of iron supplementation we are talking about. If it is labile iron, obviously you need to think about that, but I don't think the supplementation is labile iron.
Good enough. There's another one.
Any thoughts? Any thoughts on the therapeutic
Therapeutic phlebotomy of few times per year to reduce iron accumulation as a practice or anti-aging application. That's interesting. Yes.
The other day we were discussing with my teammates how iron is excreted from our body. It seems it is not really understand. We don't know how it is going to be excreted from our body, and the mechanism of regulation is not really well investigated. And it seems like blood donation seems one of the way to reduce iron accumulation. So it's like give a blood donation and then you extend your lifespan.
It's very interesting. There's another question there about ferroptosis.
Would ferroptosis affect any non-senescent cells in the body that may?
That's a very interesting question, and the answer is yes. One, it's not only senescent cells that accumulate iron. There are other cell types that also accumulate iron. For example, cancer cells, or even other type of cells, like for example, macrophages. They accumulate iron. So yes, ferroptosis might have ferroptosis as a senolytic approach might have a disadvantage because several of the ferroptosis inducers, they are not really great drugs generally. Just we showed here as a proof of principle that ferroptosis could be a potential target, and we need to get a better drug. But to me, rather than ferroptosis, just targeting the cells based on labile iron accumulation would be a much better and safer approach to kill senescent cells. And we might kill some other type of cells that accumulate labile iron, which might also be an advantage because so far what I know is like cancer cells or the M2 macrophages are accumulating this labile iron. So we might not necessarily need this type of cells in our body.
We have a question from Eric. Do you see that, Tess?
Yes. So we only see iron ions in the body and not just iron.
He's asking if that's correct.
Iron ions in the I don't understand the question. Sorry.
He says if we only see iron ions in the body and not just iron.
Not just iron.
If he's referring to the paper that I showed in the mice model, they measure total iron accumulation in the body, just total iron, not specific iron. But for the target, I would focus on the labile iron part.
We can answer Amuta, and then Eric, if you want to clarify anything, you can go ahead. So Amuta, this question.
Oh, Amuta says, what do you think of iron chelators to reduce a specific form of
iron? This is also another interesting question. If we have a very good iron chelator that passes cell membrane or that passes the blood-brain barrier to reduce iron in different parts of our body, it would be also an interesting approach to reduce. If I correctly remember, there is at least one group where they show this iron chelators might also be one approach to reduce the senescence burden, but we don't really know the details.
So Eric is saying, what I meant is, do we see any non-ionic forms of iron in the body?
Oh, okay, interesting.
I don't know. We need to specifically measure that. I honestly don't know. I don't know if other groups also measure that.
And Amit said, I think we see both total iron and iron ion with proteins like hemoglobin. That's what Amit.
Okay.
Yeah, so in our setup, what we did is we measure one total iron using mass spectrometry, just the total iron independent of the type of iron, and the other measurement using a Ceronox dye, specifically the iron 2+. So one thing we might miss is the iron 3+, which is not like really accumulated in senescent cells.
Very, very interesting. I think we don't have any more questions for now. I don't see anybody typing. Give it a few more seconds. Yeah, I think that's it for now, Tess. Thank you again for wonderful and fascinating work.